Hey guys, it's Ryder and welcome to Mall Bio Review. Today I'm going to review two papers. It's going to be a summary format geared towards students. So if you're a student like myself, I hope you'll find a digest of these articles helpful. First paper we're going to look at is about MS, and it's about the trafficking of T-cells within the brain. It's by Christian Schläger et al., and it comes to us from Dr. Flügel from Göttingen in Germany. I'm going to mainly focus on the figures, so let's just jump right into it. Figure 1. Turn to Figure 1. So Figure 1 is kind of interesting because it refutes the idea that T-cells enter the parenchyma from the choroid plexus. In fact, what they show is that by the time T-cells have infiltrated the spinal cord and the brain, there are hardly any T-cells at all in the choroid plexus, so that's out. Panel B of Figure 1 is where they show some 2-photon laser scanning microscopy images of these T-cells. So 2-photon, right? 2 photons are absorbed by the fluorophore at the same time, scanning microscopy. What they show in this panel B, the orange arrow shows the flow of CSF. They show how a T-cell extravasates from the blood vessel. It crawls around in the extracellular matrix, but then it gets detached from the ECM and is pulled by the flow of CSF. Figure 2 is when they are going to try to characterize these two types of T-cells. So what they do is they do qPCR. Panel A shows all those genes that are not differentially expressed. So among them, CXCR3, CCR5, doesn't say much to me. Panel B are the genes that are differentially expressed, and they're highly expressed in T cells in the parenchyma, but not in the CSF. Among them, interferon gamma, CSF2, that's GM-CSF, and IL-2 receptor, so that's pretty interesting. Panel C, interesting because they use NFAT, nuclear factor of activated T cells. They use a reporter for that to quantify their T cell expression, their T cell activation, and what they find is in the CSF, T cells are not activated. In the white matter and in the meninges, the T cells become activated. Panel D is a bit misleading to me. It's a bar graph, but it shows the clinical score of adoptive transfer EAE rats. So just pretend that's a line graph. They can show here that their CSF-derived cells, so those T cells that weren't activated, can still cause disease, just as well as control. Panel E, here they show that TMBP cells, but not the non-specific T cells and not T-MOG cells, which in rats apparently causes a less severe disease. The TMBP cells only are found in the parenchyma. The other two cell types are found in the CSF and the meninges. So again, it's consistent with their idea that activated T cells enter the parenchyma, those in the CSF remain unactivated. And interestingly, they also show that T-ova cells can enter the CNS if they're accompanied by specific myelin-specific T cells. In Figure 3, they prove that these T cells are not following a gradient. In fact, they follow a Brownian walk. How do they represent this data? They show that in 3A, T cells basically walk in all directions of the compass. The probability of each direction is comparable. And then they fit these data to 3 different distributions. The Levy, or they call this like the chemotactic distribution, the Weibull distribution, which is the Brownian walk, and a normal distribution. And what they find is after doing a t-test and a KS test, what they find is that the Brownian walk fits the data best, and the other two don't fit the data very well. The Gaussian distribution fits a little bit on the KS test, but not really. 3C, here they show that integrin blockade of very late antigen 4, VLA4, and LFA1, what does it do? It decreases the contact duration of T cells with macrophages. It also increases their frequency, so they move around more. It increases their velocity. So we're going to see later this kind of is their hypothesis. D, it also expands the number of cells that are in the CSF. So basically it diverts those T cells into the CSF. Figure 4, this is where they show, again, 2 photon, that T cells, once they're detached into the flow of the CSF, they can reattach, and they've got some quite nice video to prove this. And again, there's another experiment where they do this integrin blockade, and what they show here is if you pre-treat these purified cells before re-injecting them intrathecally, when the cells are treated with these integrin blocking antibodies, they are excluded from the CNS, they're enriched in the CSF. The state of activation also affects this preference. When you take TMBP cells that were from the parenchyma and inject them, they preferentially move back into the parenchyma, whereas those that were purified from the CSF move back into the CSF. And those that are in an activated state, when injected back, will go to the CNS. Those that are not activated are going to stay in the CSF. So let's go back to their abstract. In multiple sclerosis, brain-reactive T cells invade the brain and induce a self-destructive inflammatory process. T cell infiltrates are not only found in the parenchyma, but also in the CSF. How the T cells reach the CSF, what their functionality is, and how they traffic is hypothetical. Here we show that effector T cells enter the CSF from the leptomeninges during Lewis rat EAE. While moving through the 3D leptomeningeal network, they get flushed from the surface by the CSF. The detached cells display lower activation levels compared to T cells from the parenchyma. However, they were not specialized because they could still cause EAE. Integrin blockade of these cells counteracted the detachment into the CSF, and T cells floating in the CSF were able to reattach to the leptomeninges through steps reminiscent of vascular adhesion in blood vessels and invade the parenchyma. Their data, this is their conclusion, it indicates that the leptomeninges represent a checkpoint at which activated T cells are licensed to enter the CNS, and non-activated T cells are preferentially released into the cerebrospinal fluid. And they hypothesized that from there, they could possibly reach areas where antigen is available and where tissue damage is present. Our second paper is about HIV, shown to me by a friend, and it's called Directed Evolution of a Recombinase that Excises the Provirus of Most HIV-1 Primary Isolates with High Specificity. This comes to us from Janet Karpinski in the laboratory of Dr. Buchholz. They're at the Technical University of Dresden, again in Germany. Figure 1. In Figure 1, they detail their target site. It's a site in the LTR, long terminal repeat, of HIV-1. They picked it because it has similarity to LOX-P, not too much, only 30%. They call it LOX-BTR, so the target's going to be LOX-BTR. But the great thing about it is conserved with 100% match in over 90% of, well, an average of 90% of HIV isolates. Okay, Figure 2A. Here they detail their scheme of directed molecular evolution. They're going to use an approach where the target is linked in the same plasmid as the combinase They're protein. Going to they have this PEVO plasmid it contains the PBAD promoter, which is inducible, contains the recombinase gene, and it contains the target nucleic acid sequence. They're going to incubate bacteria, E. coli, overnight, and in the morning what they're going to do is digest with NDE1, the restriction enzyme. And it's designed such that if the recombinase is successful at excising this target sequence, it's going to remove the NDE1 site, right? So then they're going to digest. All of the unsuccessful clones are going to become linearized. The successful clone is going to remain circular. Then they're going to perform PCR to amplify only the circular plasmid, reclone it back into the P-evo vector. So, after a few hundred rounds of this, what they managed to do is, after that, they're going to counter-select for specificity to LOX-P, the native LOX site. So in this way, their enzymes are going to get even more specificity. Then they did a quick viability test in Effector T-cells just to make sure the enzyme wasn't toxic. And figure 2b, they have their sequencing data from this library of recombinases. So BREC is their new recombinase on top. TREE was a prior effort to make an HIV recombinase. So what they conclude from this is there are 4 hotspots of mutations, but they were reached in different ways. They're subtly different in the 2 enzymes. So panel C, you can see all 4 of the hotspots are clustered around the DNA protein interface. So there's no, what do you call that? Allosteric interactions. There's no allosteric effect. But they don't go into much detail other than that in the structure. Figure 3. Again, this bit is a little bit misleading because 3a, they show 2 gels showing, the cleavage of the target sequence as a function of the inducer, arabinose. But, and it looks like from this that BREC would not be specific at all, but in fact BREC is being used against its target and TREE is being used against its target sequence. And then in B, they're showing the specificity of BREC against the LOX, BTR, and 3 other LOX sites. And here they've chosen to use 1,000 microgram per ml arabinose. So even though earlier in the paper they said they were selecting for an enzyme that would be effective at low expression levels, here they've just gone and used the highest dose possible. So whatever. But then they do show that it has quite satisfactory specificity against LOX-P, LOX-LTR, which was the site from this older enzyme TREE, and the self-inactivating region of its own plasmid, because they're going to have to transduce this BREC on a lentiviral vector itself, so it doesn't excise that, which is good. 3C is just kind of a recapitulation. They're going to use a cell-based assay. The cells are going to turn white when the recombination happens. TREE can only recombine its own site. BREC can only recombine its own site. And then they also have CRE, the original enzyme, in there too. Figure 4, same thing as figure 3, but in mammalian cells. Quite satisfactory specificity here. And cool thing they have also is 4C, they have this primer set against the excision product, which is going to allow them to detect the excised virus. And they have good results with that too. Figure 5 and 6, I don't have too much time to get into it, but basically this is where they kind of validate this a little bit. In one N=1 HIV patient, they showed that figure 5, B and D, actually just B and C, they find that they can get the viral load down, that's part B, and they can get excision quite high, that's C, the deep sequencing. So, sorry I didn't get to figure 6, but that's all the time I've got for this time. Hope you enjoyed this episode of Mol/Biol Review, and leave me comments if I may have missed anything obvious, and thanks for watching.